Biology Part 87 Regulation of Salt. The salt concentration in plasma and tissue fluids, although indirectly affect the ADH, is under the direct control of the hormone aldosterone produced by the adrenal cortex as indicated in the figure. Aldosterone is secreted when the levels of salt in the blood falls. It stimulates sodium plus reabsorption in the distal convoluted tubules and collecting ducts. The chain of the events which leads the secretion of aldosterone begins the decrease in plasma volume, which is inevitable result of the water shortage. In the kidney, special cells lining the arterioles react to such decrease by releasing an enzyme called renin into the bloodstream. Renin catalyzes the conversion of a blood protein called angiotensinogen and angiotensin, and it is the presence of this substance in the blood which causes aldosterone to be released from the adrenal cortex. And the angiotensin also affects the diameter of arterioles and influences the brain, causing sensation of thirst. When the body suffers a prolonged shortage of salt and osmoreceptors fail to stimulate the pituitary, so less ADH is produced and greater quantity of dilute urine is formed. In this way, the body maintains osmotic pressure of its fluids at the expense of its volume. In sweating, both water and salt are lost, but often only water is replaced. The consequence may be muscular cramp caused by the drop in the level of sodium plus ions in tissue fluids below that which is necessary for the muscle reaction as well. Excessive intake of either water or salt causes an increased urine output input to eliminate the surplus. Although the human body can tolerate relatively large increase in the water, it does not have the same facility to excrete extra salt. And this is because salt must be removed in solution, and even at its most concentrated, human urine cannot carry only 5 grams of salt per liter of water. This explains that why the stranded sailor cannot survive by drinking seawater. Seawater contains about 10 grams of salt per liter, so for every liter of water obtained, it would require 2 liters of urine to be eliminated the unwanted salt. Special adaptation for osmoregulation fish. Special adaptation for osmoregulation. Let's talk about fish. The maintenance of a constant osmo concentration in tissue fluids present a problem for an aquatic organism which needs to exchange materials with the surrounding water. For example, gas exchange in fish requires a large surface area across the diffusion can occur. However, from an osmoregulatory point of view, the gills represent an unprotected area where water may be gained or lost according to the osmotic gradient between the internal and external fluids. Freshwater, Freshwater fish gain water by osmosis from their surrounding and therefore require to eliminate excess water. This is achieved by a high rate of filtration through the huge numbers of glomeruli, resulting in the large quantities of extremely dilute urine. The urine produced is mostly water because salts are very efficiently reabsorbed by the kidneys tubules. Some salt losses are made good by an active uptake of mechanism at the gills. The kidneys are not important in the excretion nitrogenous waste because this is mostly eliminated by ammonia as diffusion across the gill surface. Marine fish normally have tissue fluids which are less concentrated, hypotonic compared with the external environment, and therefore tend to lose water by osmosis. This situation can be described as physiological drought and requires efficient mechanism for conserving water. These mechanisms are entirely different in two major groups of marine fish, as described further.